Perfect. Welcome again. Today, our speaker is Abhishek Wahi, a PhD candidate in cancer biology at Purdue University in their interdisciplinary Pulse program. He's currently conducting research in the lab of Dr. Nathaniel Mabe in the Department of Medicinal Chemistry and Molecular Pharmacology, focusing on identifying and targeting epigenetic and transcriptional dependencies that drive therapy persistence in neuroblastoma. This work integrates multi-omics datasets, various sequencing techniques, and CRISPR-based functional genomics from large-scale resources such as DepMap to uncover lineage-specific vulnerabilities. In particular, he investigates how epigenetic regulators contribute to drug-tolerant persistent states, lineage plasticity, and neuroblastoma progression, and how these dependencies can be therapeutically exploited. Prior to joining Purdue University, Abhishek completed his Bachelor of Pharmacy from Delhi Pharmaceutical Sciences and Research University. He has received several recognitions, including the Lieutenant Governor Trophy for Academic Excellence in his undergraduate and the Lynn Fellowship at Purdue. We're excited to hear you speak today, Abhishek. I'll turn the share over to you.
Thank you, Haley, for this wonderful introduction. And it's my pleasure to be here. Is my screen? Let me share my screen. Okay. Is the screen visible?
Yeah.
Okay. Thank you once again, and it's my pleasure to be here as a speaker today. And thank you to the review committee for this Midwest Bioinformatics seminar series for accepting my abstract and for the invitation once again. So the title for my talk is Multi-Omics Integration Identifies USP7 Inhibition as a Targetable Dependency in the Neuroblastoma through inhibition of PRC1 activity. This is the outline of my presentation. I'll start briefly about how do we target pediatric cancers, particularly highlighting the main hallmark difference between the adult cancers and the pediatric cancers and how we approach pediatric cancer therapeutically. Then I'll give the brief background about the neuroblastoma, followed by the gap in knowledge we have. And then I'll highlight the key findings we have from this project and the conclusions and the future directions, followed by the acknowledgements. So starting with the main key hallmark for the pediatric cancer is, so as shown in this figure, pediatric cancer exhibit lower genomic alterations when compared to the adult cancer types. Only a small subset of pediatric cancer, like high-grade glioma here, reach to the mutations. This difference highlights an important biological implications. While adult cancers are often driven by accumulated genetic mutations, pediatric cancers are most commonly driven by epigenetic dysregulation and disrupted developmental programs, where cells fail to properly differentiate. To systematically uncover these targetable opportunities in pediatric cancer, we leverage large-scale resources like DepMap. And so what DepMap is, to overcome this, so DepMap integrate multi-layers of data, including genome-wide CRISPR-Cas9 screenings, and integrated with multi-omics characterization of cell lines and drug sensitivity profiling to build predictive models of cancer dependencies. This allows us to move beyond simply asking what is mutated to instead ask what is essential for the tumors to survive. Using this DepMap, various studies have found that using the dependencies values for each gene in these cell lines, when you perform the clustering of these dependencies values, we can find that this single level gene dependencies cluster around the tumor types. As you can see, that gene dependencies which are essential for the prostate cancer align in this cluster. For the focus of my talk, we'll be focusing on peripheral nervous system, in which neuroblastoma is. Along with these lines, what I want to highlight is that the dependencies of the pediatric cancers, when compared to the adult cancer, the gene ontology of those genes highlight that these cancers are particularly driven by transcription and the differentiation programs. That means the pediatric cancers often rely on transcription factors as well as the failure of the differentiation programs. Starting with the neuroblastoma, which is the focus of the talk for today. So neuroblastoma is a neuroendocrine tumor that develops from the due to the abnormalities in the development of neural crest cells. In the normal physiology, neural crest cells delaminate along this neural cord and undergo differentiation processes to either differentiate into the adrenal chromaffin cells or sympathetic ganglia. However, due to abnormalities in the differentiation processes, neuroblastoma develops, resulting in uncontrolled cell proliferation and failure in the neuronal differentiation. Neuroblastoma accounts for 15% of the pediatric cancer-related deaths. Children with high-risk neuroblastoma undergo this particular treatment strategy, which is started with the induction chemotherapy, which includes 5 to 6 cycles of chemotherapy, followed by consolidation therapy, which includes radiotherapy as well as stem cell transplantation to rescue bone marrow, and finally concluded by maintenance therapy, which includes the isotretinoin, a retinoic acid derivative for differentiation, and anti-GD2 immunotherapy. However, even after this intensive multimodal therapy, neuroblastoma patients often end with dismal outcomes, highlighting the need for the more targetable and more reliable therapeutics for the neuroblastoma patients. Neuroblastoma is often driven by feed-forward oncogenic signaling, where multiple transcription factors rely on each other to progress the tumor progression. And these transcription factors often work in coordination with epigenetic regulators like BRD4, and there are multiple examples. For example, I'm highlighting that the HEN2 transcription factor, particularly BRD4, which works in coordination with S3K27 acetylation mark, and literature has suggested there have been several epigenetic inhibitors which work by disrupting this transcriptional feed-forward oncogenic signaling, highlighting the fact that these epigenetic modifiers probably are the therapeutic opportunity for neuroblastoma. But the main difference is these epigenetic modifiers do not work as a single subunit. They work as a multiple units complex. So the question we asked is, are we missing multi-subunit epigenetic complexes by focusing on finding the most significant and significantly enriched single genes? To find such complexes, what we did is we used all dependencies data available on the DepMap and performed single sample gene set enrichment analysis using curated quorum database to identify the protein complexes that are enriched in neuroblastoma. And what we found that PRC1.1, which I'll refer as the P core in the rest of my slides, is the selective dependency in the neuroblastoma when compared to the other solid tumor lineages. We found the other complexes as well, like Saga complex, which we have published earlier. The importance of this analysis is, so when we looked at the single level gene dependencies, we looked at various PRC1 subunits are not are not come out as the selective dependencies for neuroblastoma. However, this highlights the importance of doing the protein complex level dependencies analysis. To further illustrate that this is selective for neuroblastoma, we did single sample gene set enrichment analysis using dependencies against all the cancer lineages, and we found that the neuroblastoma is the one which selectively relies on PRC1 complex for its survival. To again look at whether it's a therapeutic opportunity in neuroblastoma only, we looked at CCLE and Treehouse for the expression data for the PRC1 complex, and we averaged the expression values for all PRC1 subunits together. And we found that the neuroblastoma is selectively has higher expression of PRC1 units when compared to the other cancer lineages, highlighting the fact that higher expression of PRC1.1 complex may demonstrate the unique activity in neuroblastoma and thus could be therapeutically targeted. A lot is known about PRC2 complex, which is responsible for S3K27 trimethylation for transcriptional repression, and EZH2, which is the catalytic unit of the PRC2 complex, responsible for this deposition of the methyl group on the histone 3. There have been several literature which has published on this particular subunit to target the PRC complexes, mainly for promoting the differentiation. However, due to the limited toxicity of the EZH2 inhibitors, there is opportunity to focus on the alternative targets. And that's why we are focusing on PRC1, which is a selective dependency in the neuroblastoma. There is very less literature on PRC1. There are 4 canonical PRC1 complexes based on the CBX unit, and there are 3 PRC non-canonical complexes or variant PRC1 complexes based on the defined by the PCGF1 unit present in them. The focus of the talk is PRC 1.1. So we have ring 1AB, which is responsible for depositing ubiquitin mark on lysine 119 on histone 2A, which coordinates with S3K27 methylation to transcriptionally repress the gene programs. To validate our computational analysis, what we did is using CRISPR-Cas9 knockout, we knock out the PCGF1 as a selective dependency unit. And we found that the individual knockout of these subunits in the PRC1 complex reduced the cellular viability in the neuroblastoma cell line. Interestingly, what we found that when we knock out PCJ1, there is no change in the H2A lysine 119 ubiquitination mark, highlighting the fact that the effect of PCJ1 knockout is localized and focal, not as a global. We did a cell cycle analysis as well to look whether there is increase in the cell cycle arrest or not, and we found that there's a decrease in the S phase arrest and increase in the G0 G1 arrest, highlighting the reduction in the viability is due to the reduction in cell cycle. To understand what this PRC1 and PRC doing on the chromatin, we performed cut and run and chipseq analysis by using PCG for the PCJ1 using two different antibodies to gain the accuracy and identification of the peaks of the binding of PCJ1 on chromatin. And what we found that we found two different subclusters, and here we have this is the cut and run, and for EZH2 we have chipseq, calibrated chipseq. So for PCJ1 and ring 1B is defining the PRC1 complex here, and EZH2 is defining the PRC2 complex here. And these are the histone marks defined by the PRC1 complex and PRC2 complex. So what we first found is there is a subcluster of the peaks which is PRC2 co-bound. That means the PCJ1 and EZH2 are present on the same chromatin sites. Interestingly, what we found that there is a subset of peaks which are enriched for the PRC1 only, not for PRC2, which can be defined by the intensity of the peaks in these two lanes, not in the EZH2, which defines for the PRC2. And along with this, what we found that the PRC2 co-bound and the PRC2 independent peaks are enriched for the promoter sequences, that highlighting their effect on the transcriptional level. We looked at the expression of these peaks as well, that the PRC2 co-bound are low expressed when compared to the PRC 1.1, which is the independent peaks here, are highly expressed in comparison to the PRC2 co-bound peaks, which highlight the literature that the PRC2 co-bound sites are actually responsible for transcriptional repression. To gain further understanding of what PRC2 independent peaks are doing, we looked at the motif analyses, and we found that several of the transcription factor came out as the top hits, which are responsible for maintaining the oncogenic signaling in neuroblastoma. But to gain the mechanism and gain the understanding of what PCGF as a target is doing, we knock out PCGF1 and perform the bulk RNA sequencing to understand the transcriptional consequences associated with PCGF1 knockout. And we found that the quantification of the DSEQ2 analysis highlighted that there's a preferential upregulation of the genes in comparison to the downregulation of genes when PCGF1 is being knocked out. We did an overlap analysis to find out the selective genes which are being upregulated, and we found 165 genes in the here. And we found that several of the genes are responsible for the neuronal differentiation as shown by this gene ontology analysis. To gain further understanding, is it the true gene neuronal differentiation program? We performed gene set enrichment analysis and found that the several of the neuronal differentiation pathways are the top hits. To understand the epigenetic consequences, we looked at the PCGF1 knockout by using ChIP-seq. And when we knocked out PCGF1, there is a decreased binding of PCGF1 on chromatin, along with decreased binding of the ring 1B, which is the catalytic unit of PRC 1.1 complex responsible for the deposition of H2AK lysine 119 ubiquitination. So when PCGF1 is knocked out, there is decreased binding of ring 1B as well and decreased ubiquitination, highlighting the fact that transcription is being upregulated because of the decrease in ubiquitination. But there have not been any inhibitors for PRC1 or PCGF1 so far. So we were looking for translational approach. So we looked at, so there are ring 1A, ring 1B inhibitor developed by University of Michigan in some micromolar range. However, the ring 1A and B unit is present in all of the PRC1 variants. So we were looking for sub-complex therapeutic opportunity. So we looked at USB7, and if people are familiar with the field for the USB7, USB7 is mainly responsible for the turnover of the P53 by inhibiting MDM2, which is responsible for the degradation of P53. So when we looked at the correlation analysis using the depth map, we found that the USB7 strongly correlates with P53, which is known with the literature. And when we use the USB7 inhibitor, which was provided to us by Dr. Sarah Burlage from the Dana-Farber Cancer Institute, we found that the P53 intact cell line is more responsive to that USB7 inhibitor when compared to the P53 mutated. And this can be recapitulated in vivo as well. So we were surprised and we questioned whether P53 pathway dependency is so dominating that it is dominating the other pathway dependencies. So what we did is we generated a TP53 dependency CRISPR score by averaging the dependency score for these three, and we found the cell lines which are highly dependent on P53 and which are not dependent on P53. So using the cell lines which are not dependent on P53, we did correlation analysis again. We found at the PCGF1 as the strongest hit. Sorry for the animation here. So PCGF1 is the strong dependency hit here, highlighting the fact that USB7 could be a co-therapeutic target for targeting PCG1 or PRC1 complex here. And that's what we observed that the piece this is and we used that USB7 inhibitor, which I showed in the previous slide, and we found that there is a reduction in the viability when compared to the DMSO control here in the neuroblastoma cell line. And this result can be recapitulated in the tumor volume as well in vivo. And there is no difference in the body weight of mice, highlighting that the drug is safe to use. And as the biomarker, we looked at the SOX13, which is the one of the top hit from our bulk RNA sequencing from the previous slide I showed, that it is being upregulated in vivo as well, which is responsible for the neuronal differentiation in the neuroblastoma. To understand whether these transcriptional epigenetic consequences mimic the PCGF1 knockout, we did transcriptomic study and epigenomic study using USP7 inhibition, and we found that this is again preferentially upregulated for the transcription. And for looking at whether this PCGF1 inhibition by indirectly by USP7 inhibition, we looked at the GSE analysis by looking at the PCGF1 core gene sets, which we identified using PCGF1 knockout, using the overlap analysis of 165 genes. And we found that the USB7 inhibition is positively enriched for the PCGF1 core gene set. That highlights that there is a strong correlation between PCGF1 knockout as well as USB7 inhibition. Again, looking at the GSE analysis, there is a strong upregulation of the neuronal differentiation pathways, highlighting that there is not indication of apoptosis, not cell death, but the reduction in viability is due to the terminal differentiation. To understand the epigenetics, we did ChIP-seq again, and for lysine 119 ubiquitination, which is responsible for transcriptional repression, and we did see that there is a reduction in the ubiquitination mark. As you can see that there is a subset of peaks which have decreased in the ubiquitination, and there is a global reduction in the ubiquitination as well. We integrated our epigenetic as well as the transcriptomic analysis by integrating our RNA expression log to fold change from RNA expression and log to fold change in the ubiquitination mark. And we found that when there is decrease in the ubiquitination, the genes are being more expressed, and we found that 931 genes are being more upregulated. We looked specifically at PRC 1.1 and PRC 2, which I highlighted as the PRC 2 co-bound sites, and we found that these are the particular peaks which are being upregulated. So that means these peaks come from the peaks which are not PRC 2 independent, these are PRC 2 co-bound sites. Gene ontology analysis using these consensus gene set, we found that these genes are mainly regulated for neuronal differentiation, and for the C2 analysis for C2 gene sets from the MSIC-DB, we found that these are the genes associated with PRC1 and PRC2 co-bound sites. It's highlighted that S3K27 methylation here. And there are also different differentiation related pathways, highlighting the fact that the USP7 is increasing the inhibition, increasing the upregulation of the differentiation pathways. To understand the mechanism, we did global proteomics as well as ubiquitomics at short time points as the longer time points as well. And the analysis of these experiments showed that there is a reduction in the PRC1 complex sustainably at shorter time points as well as the longer time points. We looked at the individual subunit values as well. We found that several of the PRC1 units like KDM2B, USP7, PCGF1, and BCOR are the most downregulated proteins. Interestingly, we found that PCGF6, which is another variant PRC1 complex, is also downregulated. We looked at the dependency for PCGF16 as well in the map data, but it was not selective dependent. To understand whether this is true, we looked at the Western blots as well using the XL177, which is a USP7 inhibitor. There is indeed the reduction in the PCGF1 protein level and the PCGF6 as well. Interestingly, there is no change in the canonical PRC1 and no change in the PRC2 complex. That means it is selective for PRC1 only. Since there is reduction in protein, we wondered whether it is because of the decrease in the transcription. So we looked at the qPCR for the PCGF1. There is not much significant change in the transcripts for PCGF1. It highlights that it could be possible that there is a difference in the stability of PRC1 complex rather than there is a changes in the transcription of the PCG1. We used MG132, which is a proteasomal inhibitor, and MLN7243, which is even ligase inhibitor. So these inhibit activity of a ubiquitin substrate to inhibit the degradation of any protein. So when we used our XL177A, we can see that there is reduction on the proteins. However, when we use our ubiquitin proteasomal inhibitors, there we can rescue the phenotype here, highlighting the fact that this is because of the reduction in the stability of PRC1 subunits, not changes in the transcription for that particular proteins. We looked at the ubiquitinomics data, and we found that there are several peptides for ring 1A, which is one of the member of the PRC 1.1. Specifically, the lysine 261 is highly ubiquitinated. And this particular lysine is responsible for the interaction between RYBP YAF2 and ring 1AB, which is responsible for maintaining the PRC 1.1 stability for depositing H2A lysine 119 ubiquitination mark. So what we hypothesize from here is that when USB7 inhibitor is being provided in the cells, that ring 1B is retaining its ubiquitination mark and not able to deposit it on the histone. We performed co-IP analysis to understand it much better, and what we found that when we use our USB7 inhibitor, that in the presence of no inhibitor, there is a strong interaction between PCGF1 and ring 1B, which is present here, like which is retaining the ubiquitination here in the presence of inhibitor. However, when the inhibitor is given, the interaction is being lost with ring 1B as well as YAF2, highlighting that fact that it is due to the disruption of the stability of the complex. Overall, the mechanism we concluded here is the USP7, which is the inhibitor for the which is in normal physiology, is preventing the degradation of the ring 1B. Is in that scenario, ring 1B is able to interact with YAF2 and RYB and coordinating with USB7 to deposit the lysine 119 ubiquitination and preventing the transcription of the neuronal differentiation genes. However, when USB7 is inhibited, ring 1B is no longer able to deposit the lysine 119 ubiquitination because of the instability of the PRC 1.1 complex. And that's why the chromatin is being more opened for the neuronal differentiation genes upregulation. The conclusion is that PRC 1.1 is critical epigenetic dependency in neuroblastoma, which maintains the undifferentiated tumor state. And when it is inhibited indirectly by USP7 inhibitor, it can promote the differentiation of the neuronal differentiation and suppress tumor growth. I would also like to highlight the future directions that we found that USP7 inhibition, which is the inhibition for B core complex, also known as PRC 1.1, we want to see whether we can apply this therapeutic opportunity to other pediatric cancers or not. Specifically, we are looking for B core mutated osteosarcoma patients because we recently got those samples from our collaborators. So we are very excited about this project as well. And from our proteomics data, we found that the PRC 1.6 was also the downregulated abundant protein in the presence of USP7 inhibition. So we want to understand what other variant or canonical PRC1 complexes are doing in the presence of USP7 inhibition. And specifically, we also want to understand the PRC2 independent sites, which are responsible for maintaining the transcription factor oncogenic signaling. Finally, I would like to acknowledge everyone. First of all, I would like to acknowledge Dr. Nathaniel Mayer, my PI, who, for providing me the opportunity to contribute the first piece of the project in our lab. And I will also like to thank Xing Liu from Purdue and Dr. Stegmeyer from Dana-Farber for contributing to this project. Also, I would like to acknowledge Gerard Marto and Sarah Burlage for providing USP7 inhibitor and for proteomics and ubiquitinamics study. I would also like to acknowledge my funding sources and Purdue Institute for Cancer Research and RCAC Purdue and Bindley Genomic Core Facility and Bioinformatics Facility for providing me access to Nigishi. And thank you once again to the Midwest Bioinformatics Seminar Series for providing this opportunity to present my work here. I'm happy to take any questions.
Thank you. Very nice talk, Abhishek. Thanks for sharing
Thank you.
that with us. Anyone is welcome to ask a question. You can come off mute and ask it directly. You can put it in the chat, and then I will read it out if you'd prefer to do it that way. So let me open the chat so I can see that. Um, and I'll give folks a minute to write. So I can start us off with a question. I am not a cancer biologist, so this might be a little
It's
bit
okay.
of an uninformed question, but it was interesting to hear you talk about the differences in pediatric cancer versus adult cancer, both from a causal perspective and then also following that, how you have to treat it or target it. But I was wondering if you could speak a little bit about whether you think that this target would also apply to adult cancers at all, if there are cases that are still sort of more transcription factor regulatorily um, governed and not just accumulation of mutation. Like how is there some overlap in this target for adult cancer as well?
There have been several literature regarding, particularly for USP7 inhibition in adult cancers as well, but it is not due to the regulation of epigenetic, but it is mostly by apoptosis, increase in apoptosis, which is the primary function of the USP7, which I highlighted here, that it inhibits MDM2 and prevent the P53 decay. So when USP7 is inhibited in other cancers as well, it will increase the P53 dependent apoptosis. So it could be possible that USP7 inhibition can be applied to other cancers as well. Yeah,
Thank you.
thanks.
I'll go next, if there is no one asking question. Um, yeah, so I'm also not a cancer biologist, but interested in the bioinformatics method. When you integrated your ChIP-seq, cut-and-run, and RNA-seq data, how did you handle the peak to gene assignment? Did you just use the nearest TSS approach,
Yeah.
or did you use something like more sophisticated?
So for this particular integration, what I did was I looked at the promoter sequences plus to minus 3 kilobases near the transcription start site only.
Okay, so the
And then I
closest tier.
calculated the area under curve for that peaks only.
Okay, okay.
Yeah.
Um, so I might have missed it. Did you use any IC-based methods to link these
No,
things, or no? Okay, good.
no. We are interested in that and we will be doing it in future.
Yeah, I was wondering that would be like more sophisticated way to get all these
Yeah, yeah.
things.
To gain the understanding of long range interactions of the enhancers with the
Yeah,
promoters and all those things. Yeah. We are interested in that for sure.
very cool. Thank you.
Yeah.
I think a little bit more along the lines of the methods.
Yeah.
You highlighted at the beginning the um, sort of inability of single gene expression or enrichment level analyses to pull out the influence of entire like complexes.
Yeah.
So when you're switching to try to target looking at complexes that are involved, are you're starting with hypotheses about complexes that you want to look for so that you can pull out their signal. Were there other um, sort of like protein complexes that you had included in the hypothesis that were sort of interestingly not part of the mechanism that
Okay. Yeah, that's a good question. So we can see that there are several points along with PRC 1.1c here. So one of them is a saga complex, which is responsible for lysine acetyl transferase activity on histones. There is already a publication by my PI only during his postdoc work on that. And there is PRC2 complex, which is responsible for S3K27 trimethylation. There is published literature on that as well. So
okay.
that's why we specifically focus on PRC 1.1.
Mhm.
Yeah.
Go next. For the depth map codependency analysis, how many cell lines were you working with? Like, did you have to filter some of these lineages, or did you use all of them?
So for let me- so this was for all cell lines and
All cell
available on
lines.
that map. Yeah,
Oh,
yeah, for all
so
cell
that's
lines. And this one was for all cell lines, but selectively we were looking for the neuroblastoma. So this was like two-class comparison effect. Any other questions? Question?
um. Oh, sorry.
What would you say has been the most technically challenging part of this research?
What's your question?
What would you say has been the most technically challenging part of this research?
Most challenging part of getting results from the code. Okay. Most challenging part, if I would have to highlight, I guess it just need to do multiple times to do the experiments, but you will eventually get the results. That's what it is, I would say. But yeah, for understanding the mechanism, we had to do a lot of literature review to make more sense that since there was not much of the decreased ubiquitination for the PCGF1 proteins, even though we were looking at the reduction of the PCGF1 with the USP7 inhibition, we were still getting the ubiquitination on our ring 1 peptides. So we were surprised, okay, what could be the possible mechanism? Then we looked at the structures and then it eventually makes sense. Okay, this could be one of the possibility of the mechanism.
So USP7 inhibition is essentially like doing all. I mean, doing the terminal differentiation
Yeah,
thing. Like, how can you use this for like? Uh, I mean,
for
like to
cancer.
come up with some treatment, right? Like, how can you use this knowledge?
Okay, so for neuroblastoma and even for other pediatric cancers, since these are the cancers which are responsible, which develops during the developmental processes, it is important to prevent their growth as well as to promote their differentiation so that they can retain their normal activity as well. So even so, for it is very rare for particularly for neuroblastoma to get P53 mutation. So for even if the patient is P53 intact, we can promote neural differentiation and the patient is P53 intact, USP7 inhibition will increase the apoptosis of the cancer cells as well. So it's like hitting 2 points at the same arrow. But for the neural differentiation specifically, we have retinoic acid in the neuroblastoma treatment, which I mentioned during the beginning. Let me pull up that slide as well. In the maintenance therapy, we include isotretinoin, which is the derivative of retinoic acid for promoting differentiation only. That is the part of clinic regimen as of now. But that also has some limitations, so that's why there's need for more better opportunities.
Very cool. Thank
Yeah.
I agree. Very cool. Thank you so much, Abhishek, for sharing your work with us today.
Thank you.
Like the rest of the seminars for this series, this will be available as the recording of this will be available linked from our website. And we hope that you join us next week as well. Thank you.
Thank you.
Great presentation. Thank you.